Very good morning, all of you. I'm Satish Kumar Pandey, working as Assistant Professor in the Department of Biotechnology, Mizoram University, Aizawl. Today I'm going to discuss ELISA technique. ELISA is an immunological technique and detects antibody or antigen present in the blood or serum sample of the patient. This talk is divided into following content: introduction, basic principle of ELISA, material required for ELISA, type of ELISA, and an application part. Now we move to the next slide, that is the introduction of ELISA. Enzyme link immunosorbent assay, also known as enzyme immunoassay, is a biochemical technique used mainly in immunology to detect the presence of antibody or an antigen in sample. ELISA stands for enzyme link immunosorbent assay. I start this lecture with an example of how this test will be performed. In certain conditions, let's assume a person who has an infection in the laboratory, in his serum specific antigen are present to rule out or rule in the presence of certain microorganism. In another word, enzyme link immunosorbent assay is an immunoassay technique involving the reaction of antigen and antibody in vitro. ELISA is sensitive and specific assay for the detection and quantification of antibodies. ELISA tests are usually performed in microplate. ELISA plate are also known as a polystyrene plate, which will positively bind antibodies and protein. The binding and immobilization of reagent makes ELISA simple to design and perform. Having the reactant of ELISA immobilized to the microplate surface enables easy separation of bound from unbound material during the assay. This ability to wash away non-specifically bound material makes the ELISA a powerful tool for measuring specific analyte within a crude preparation. First ELISA test was developed for HIV virus in year 1971. The term ELISA was first used by Ingwell and Perlman. Why known as enzyme link immunosorbent assay? Number one is antigen of interest is absorbed on plastic surface, that is sorbent. Number two is antigen is recognized by specific antibody, that is a primary antibody known as immuno, and primary antibody is recognized by secondary antibodies which has enzyme attached, that is known as enzyme linked. And fourth is substrate react with enzyme to produce product usually colored. Now we move to the next slide. Here we discuss about the principle of ELISA, how ELISA works. Enzyme link immunosorbent assay principles are very similar to other immunoassay technology. ELISA rely on a specific antibody to bind the target antigen and detection system to indicate the presence and quantify antigen binding. In order to maximize the sensitivity and precision of the assay, the plate must be carefully coated with high affinity antibody. In another word, enzyme link immunosorbent assay combine the specificity of antibody with sensitivity of simple enzyme assay by using antibody or antigen coupled to easily assayed enzyme. ELISA can be provide a useful measurement of antigen or antibody concentration. ELISA can be used to detect the presence of antigen that are recognized by an antibody, or it can be used to test for antibody that recognize an antigen. Following materials are required to perform ELISA. First is ELISA plate, also known as microtiter plate or microplate, antibody, that is a primary antibody or recognition capture antibody, washing buffer, blocking reagents, secondary antibodies, substrate and stop solution. In this slide we discuss about the ELISA protocol. In general, ELISA is 5-step procedure. First step is coat the microtiter plate well with antigen. Block all the unbound site to prevent false positive result. Third is add a primary antibodies to the well. Fourth step is add secondary antibody conjugated to enzyme. Fifth is a reaction of substrate with enzyme to produce a colored product, thus indicating a positive reaction. There are several types of commonly used methods of ELISA. ELISA is basically divided into 4 different types. In case of direct ELISA, for direct detection, antigen coated to multi-well plate which is detected by antibody that has been directly conjugated to an enzyme. This detection method is a good option if there is no commercially available ELISA kit. In other way, in direct ELISA, the antigen is immobilized to the surface of the microtiter plate and detected with an antibody specific for the antigen. The antibody is directly conjugated to HRP or other detected molecule. Direct ELISA have the overall easiest setup. In direct ELISA, the direct ELISA detect the binding antibody using a second conjugated antibody. In general, the first few steps of direct ELISA are followed, including immobilization and blocking. A detection antibody is added, but unlikely the direct ELISA, this antibody is not conjugated. Second antibody conjugated to an enzyme is then added to detect this first binding antibody. Another washing step procedure, the addition of substrate and stop solution. The signal is then detected using a plate reader. The use of secondary or tertiary antibody for detection can lead to a signal amplification, but also leads to longer and more time consuming and more complex procedure. In sandwich ELISA, the capture antibody is absorbed onto the ELISA plate as opposed to the antigen. Additionally, antigen is recognized by 2 antibodies, one capture and one detection, thus forming a complex like a sandwich. Briefly, after washing and blocking the immobilized capture antibody, similar containing antigen is added to the plate. The detection antibody is added that will bind to another region of the sample molecule. This antibody may be directly conjugated to an enzyme. If it is not conjugated, a second antibody conjugation to an enzyme is used to detect the first antibody added, followed by short incubation and addition of the stop solution. The signal can each would be typically measured on plate reader. In case of competitive ELISA, also known as an inhibition or blocking ELISA, measure the amount of analyte in sample by quantification of its interference with an expected signal. Competitive ELISA can be performed in 2 ways. The microplate can be coated with an analyte or with an antibody specific for a target analyte. A target analyte that has a detection moiety can be added competing for binding site on antibody. A low amount of binding could indicate a high level of target analyte in a sample. The signal inversely correlate with amount of analyte such that if the concentration of the target analyte is high, then the reference signal is diminished through its competitive binding to a limited amount to label antibody. In another multiplex ELISA, multiplex ELISA enables a researcher to assess the level of more than one protein target at a time, and it is typically used to determine the expression of multiple cytokines simultaneously. Other molecule, unlike traditional ELISA format, multiplex ELISA method utilize one or more of the following techniques: flow cytometer, fluorescence, chemiluminescence, and electrochemiluminescence. Flow cytometric analysis is the most commonly employed multiplex ELISA method and is performed using bead conjugated antibody. Typically, a great number of cytokines can be measured at a time by use flow cytometry and bead conjugated antibody compared to other antibodies. Application of ELISA. ELISA test to detect various kind of disease such as dengue, malaria. ELISA test also be extensively employed for in vitro diagnosis produced for a It is used for immunoscreening. It is also used for food safety, and it is ELISA is also used in therapeutic drug, that is pharmacokinetic, anti-drug antibody, and host cell protein. It is mostly involved in agriculture science, testing of soil, water, seed, composite manures, biosolid, and plant tissue sample. And it is routinely used for diagnosis of medical lab. Thank you very much.